Okay, our next pathway what's called the transforming growth factor beta pathway, or TGF-beta. Now, what is TGF-beta? It's a big, diverse family of growth factors. There's many different members and subfamilies within it. They're used in numerous different developmental events. More on that in just a little bit. They're often called TGF-beta. Now, there's also a TGF-alpha, which is a different and single growth factor. But this is a very diverse family of growth factors, widely used in all kinds of developmental events. Now, what's the term transforming mean? In cell biology, when you transform a cell, you are turning a cell into something that resembles a cancer cell. Okay? So, if you treat certain cells with TGF-beta, they take on characteristics that are more what you might see in cancers. Doesn't necessarily mean they are really nasty, full-blown cancer cells, but they do take on some characteristics. Okay, so they first found, okay, it turns certain cell types and they start acting a little bit funky, a little bit cancerous or whatever. But obviously the purpose of this family is not to generate cancer in various organisms. It has many different developmental roles. We have all kinds of different members of this family. The classical TGF-betas, there's a wide range of those. There are family of proteins called the activins. They play important roles. The activins play important roles in generating the beginnings of the central nervous system. Okay. There's something called VG1, first discovered in frog eggs, and found in other organisms, different versions found in other organisms too. It's called VG1 because it's found in the vaginal or bottom part of the embryo, or rather of the frog egg. And remember, frog eggs are kind of pigmented up here, and they're clear down here. This part is the dorsal part, here's the ventral part for the most part. And what happens, if you look at VG1, it's concentrated almost exclusively to the very bottom of the ventral pole. And it plays a role in generating the ventral, primarily endodermal structures. So it helps to set the dorsal ventral axis of the embryo. Okay. Another member of these are called the bone morphogenic proteins. Or BMPs for short. With a name like that, it obviously says that at least some of them play important roles in developing and organizing the skeletal system. In other words, why your leg bone looks like that and your ribs look like something else. Morphogenesis, generating form. Okay, well, indeed, some BMPs do that, but there's many other BMPs that play all kinds of other roles that have nothing to do with bone. So that's another important family that's all in this big TGF-beta superfamily. So many different developmental events, from early events like setting the embryonic dorsal, like helping to set the embryonic dorsal ventral axis, organizing the skeletal system, certain types of organ formation, dorsal ventral part of the neural tube, in other words, saying what part of the neural tube is the top of your brain, what's the bottom of your brain, that kind of stuff. You see members of the TGF-beta family involved. So, widely used in many different developmental processes. Okay, now, what's their signaling pathway like? Okay, well, these guys are very different from what we've seen before because they do not use any G proteins at all as intermediaries. Instead, what we're going to do is activate, phosphorylate and activate a broadly used set of transcription factors. So we're going to occupied receptors ultimately activate a family of transcription factors that do all kinds of different things in terms of generating changes in gene expression. Okay, so let's look at the pathway here. Now it turns out there's not just one receptor for this, there's actually two different types of receptors that bind the TGF-betas. The receptors are fairly similar, but they're distinctly different. So we have something called a type 1 receptor and a type 2 receptor. The type 2 receptor binds the growth factor fairly strongly. The type 1 receptor on its own binds it very poorly. Okay, so what's going to happen here? First of all, we're going to have a type 2 receptor.
Which one binds tightly?
The type 2. Okay, it binds the TGF-beta. Now, that causes a structural change in the type 2 receptor, or it can now bind to the type 1 receptor, cause it to change its structure, and then also bind to the growth factor. Okay, so now once again we've got two receptors binding to each other. However, it's not identical receptors. It's a type 2 and a type 1 receptor. Now it turns out that both of the receptors, once again, are protein kinases. But they're not tyrosine kinases. They don't phosphorylate on tyrosines. They're the regular kind that stick phosphates on the amino acids serine and threonine instead. Okay, so what happens? Once we have this complex assembled, the type 2 receptor then goes and phosphorylates the type 1 receptor. And the type 1 receptor's kinase activity is now activated. So the type 2, when this thing assembles together, the type 2 kinase is turned on. The type 1 kinase is turned on when it gets phosphorylated by our type 2 receptor. So now this guy's an active kinase as well. Now its target, which it binds and phosphorylates, are a family of transcription factors called the SMADs. Now it's an acronym for something, I have no idea what it stands for, but phosphorylates this family of transcription factors. Ordinarily, these proteins are in the cytoplasm, but when they get phosphorylated, they change their structure, and it opens up and exposes a nuclear import sequence. In other words, a sequence of amino acids that's a ticket into the RAN pathway that gets you through the nuclear pores and into the nucleus. So now, these guys, which are ordinarily cytoplasmic, go into the nucleus. Okay, now, these guys then will bind to additional version of the family called SMAD4, which always stays in the nucleus. It's often called co-SMAD is another term for it. This guy stays in the nucleus all the time. And this complex, sometimes with other transcription factors, is then going to bind to certain enhancers and regulate gene expression in many different kinds of genes. Now what we see here is many of the genes that are involved in development, developmentally important, tend to be genes that start producing ventral and endodermal type tissues. That tends to be fairly common, not universal with these guys, but fairly common here. Okay, so what we see here is something kind of a lot different. We don't have anywhere near as much amplification as we used to, but of course we can phosphorylate a number of these guys. Here's our amplification step. There are no G proteins involved in this pathway whatsoever. That's unusual too. So we have a much more direct route to activating a set of transcription factors that lead to important developmental events. Yes?
Okay, so after the type 1 receptor is activated by the type 2, it's going to phosphorylate and activate the SMAD.
Yes.
And then that now has the ability to go to the nucleus.
Right.
It binds to that thing.
Yes.
And then what does it do?
Then it binds to DNA of various enhancers and activates them, or shuts them off.
Okay.
Okay. All right, so here we are basically directly activating widely used family of transcription factors. Okay. Well, that covers several aspects of some major signaling pathway. Now we're going to look at something else that also, there are a few more pathways we'll get to later on, but we're going to look at something else that also plays crucial signaling roles, even though it took a while for that to be realized. And it also plays many other important roles, both at the cellular and at the organismal level.